Good afternoon, everybody. Welcome to class. How's it going? Hi, everyone. Hello. Afternoon, Emily. Oh, who was that? Hello. Good afternoon. Joseph, how's it going? Excuse me. Afternoon, Arlena. Good afternoon, Dominic. There we go. Alrighty. So it's 12:20. We'll give everyone a couple of minutes just to kind of get in and sorted, and then we will get our lesson started. I'll share with you my schedule for the day. Or the plan for the day. You have access to that document as well in the Unit 1 Diversity Outline. But as always, we'll start our day with it up just to kind of talk about it and generally ask questions if there are any for you all. We're not on that day. We're on this day. We're on day 1 of prokaryotes and bacteria specifically. We will talk about today. On Wednesday next week, we'll do part 2 where we finish up bacteria and talk about archaea. So I'll just give everyone a couple more minutes before we get started, and then we can take a look at the note. I'll be doing it a little differently today because the PowerPoint slide, while informative, does not have all the information that I want you to get down on paper if you are following along. So I will do it a little differently today, probably moving forward with the note, just so I can ensure that you're filling in the things that I would like you to fill in if you're following along that way. Good afternoon, Natalia. Alrighty. Let's see, where are we numbers wise? Kiara, good afternoon. Hello. Alrighty. We're almost at our number. We're missing just a couple of people. I'll give everyone 1 or 2 more minutes. For those of you that would like to follow along today, we will be working through this document. I'll put it up on screen in a second. I just have the schedule up there, just so those of you who are looking to see what lesson we're opening up and what we'll be working with, you can see it from there. But otherwise, this is the document that we will be working with today. Lesson 5, prokaryotes, bacteria, and archaea. I will do the first half today, and then I'll do the second half all the way on next Wednesday. So it should be fine though, because we get into the meat and potatoes, so to speak, of it today. So I think we'll be okay in terms of what we are studying or what we are practicing with and learning today. And then I'll see if we can't finish a little bit earlier, so that way you have some time to work on your lab activity towards the end of class. All right, let me just quickly take attendance. Emily, Tom, Helen, Jaden, Janice, Jonathan, Joseph, Ken, Kiara, Maddie, Andre, Luca. I think I found everyone that I'm missing. No, I haven't. Maddie, Megan, Lena, Natalia, Tammy, Vanessa. I'm sure I'm missing someone else. second. All right. Well, oh, there we go. Found them. Haha. Okay, so we can get started once this last person joins, and I'm hopeful that the other people who have yet to come will join us. But those of you following along today, this is the lesson we will be working with. Oops. Go back to that. And then let me just fix that. Alrighty. So we will start by talking about prokaryotes and eukaryotes once I share my screen with you. And we're going to talk about the general structure as well as their different classifications and what we use to kind of describe and define them. Because prokaryotes, specifically bacteria, which is what our focus is today, they're quite diverse and they have a lot of major characteristic differences from each other that allow for them to kind of behave in specific ways that are both good and bad for humans and other animals as well. They are quite fantastic in terms of how diverse they are, how they survive, and the general structure and idea of them, which we focus on today, will be, my hope is that it will be, if not fascinating, informative for you. So the key component here that I want to discuss with regards to prokaryotes is the main structural differences from eukaryotes. So just from a review from what you've learned in grade 9 biology or grade 9 science, prokaryotes are never going to have a membrane-bound nucleus, meaning there's no actual functional nucleus like there are in eukaryotes. They will always be unicellular microscopic organisms, and they encompass the domain of bacteria or eubacteria and archaea or euarchaea. So it's important to recognize that this domain that falls under the kingdom of prokaryotes, or the domains that fall under the kingdom of prokaryotes, are archaea and bacteria. Like I said, we will focus on the bacteria for today and then archaea on the next time that we talk about this content. So why bother studying bacteria? What is the point of understanding how bacteria work? Well, I'll talk about some of the functional aspects with regards to them as we move through this lesson. But just in general, with regards to habitats on Earth and the planet itself, there are millions upon bacteria on and in us. So on our skin alone, there are tens of millions of bacteria that live there. Within our gut, there are tens of millions of bacteria that are in there as well. And so it's one of those, who am I missing? Sorry, 1 second, folks. Oh no, I'm the person. Haha, never mind. So these millions of bacteria that live on and in us, the bacteria inside of our gut, inside of our ears, inside of our mouths, inside of our eyes, nose, every orifice that you can find on the human body, it is inundated with bacteria in some way, shape, or form. The interesting component with regards to it is sometimes they're quite helpful. And in fact, we need to utilize them and have them inside of us in order to survive. And I'll talk more about that later. With regards to archaea and the specific idea that revolves around what they are functional with, they are really, really good at living in extreme environments. Very, 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 very high heat, very, very, very low heat, high salinity, high pressure, all those extreme environments that you can think of, archaea are quite good at living there. So I'll talk more about them next week when we focus on them on Wednesday's lesson, but they are incredibly good at living in environments that do not favor normal microscopic organisms. All right, questions about just the general introductory components of them? Okay. So the next concept that, if you're following along and writing in as we go, the next concept that I'd like you to pay attention to is that the idea of where prokaryotes and how they function, where prokaryotes are and how they function. So like I said, there are millions of bacteria living on and inside of us, but they also vastly outnumber any other organism that exists on this planet. We've only classified approximately 1%, let me just clean that up a little bit, 1% of them because they're incredibly difficult to find and access. In the water alone, in the ocean alone, we have no idea, just hearkening back to that video that Dr. Alvarado, that we saw with Dr. Alvarado on the first day, We have no idea what exists bacteria-wise in some of the deepest parts of the ocean because we've yet to actually travel there and collect samples. So 1% of the known bacteria that we have actually, or think that actually exists, we have actually classified and we have actual samples of. So it's quite a daunting task in terms of understanding and categorizing all of these microorganisms. There are two main types of bacteria that we will speak about in this class. And the first is that they are what's called pathogenic. So bacteria that are pathogenic can be quite harmful to us. Specific diseases like strep throat, which I'll talk more about later, cholera, and food poisoning that happens as a result of a very harmful E. coli strain, those are all pathogenic, meaning if we come in contact with that bacteria under the right circumstances, it will make us sick. So strep throat, if you've ever had strep throat, which is a throat infection as a result of a specific bacteria. Cholera, same thing. And food poisoning, if you've ever had undercooked, usually it tends to be with regards to meat, but if you've ever had undercooked meat, you can get food poisoning from that. There are bacteria, however, that are very helpful and play a very important role in day-to-day life for humans. And that bacteria which is helpful, they allow for our ecosystem, both human ecosystem and ecosystem around, to function the way with which we need it. Yeah, usually E. coli is as a function of undercooked meat. Sometimes it can be from vegetables that haven't been washed properly, but usually it tends to be meat that is, for lack of a better word, not the highest quality, or it's undercooked severely as a result of user error usually. Alrighty, so these helpful bacteria can include, but are not limited to the examples that I discuss here. So the first concept that I want to talk about with regards to helpful bacteria is probably the most important one. It's often overlooked, but it is indeed the most important idea of a helpful bacteria. And it's the bacteria that help to fix nitrogen and allow for plants and humans later on to absorb that nitrogen. Without them fixing nitrogen into soil or into nutrients, we would not be able to have atmospheric nitrogen. We would not be able to utilize that nitrogen for our DNA and proteins, and life itself would cease to exist the way we know it. So these helpful bacteria that fix nitrogen from the atmosphere into nutrients and out of nutrients are quite good at keeping us alive, for lack of a better word. When it comes to bacteria that go through what's called photosynthesis, we'll spend a lot of time talking about photosynthesis throughout this course, but photosynthetic bacteria will produce oxygen and sugar in many different ecosystems, specifically ones in the ocean. These photosynthetic bacteria are the main food source for fish and other organisms that live in water. And so without it, the food chain would be severely disrupted, and we would see some other issues arise as a result. Alrighty. And then to move on to a more relevant topic with regards to humans, we as a species need to have a very healthy gut flora of helpful bacteria, because these helpful bacteria allow for us to produce and absorb vitamins K and B12, which are incredibly important for energy production within our cells. And so without this bacteria inside of our large intestine, basically activating and creating our creating the ability for our bodies to produce and absorb this vitamin K and B12. Without these bacteria, we would never be able to access these vitamins. And as a result, you could eventually die from a deficiency in vitamin K and B12. So it's important to recognize that in the context of, just how important it is to have access to that. And then lastly, the last two, sorry, I should say, because there's still two more. Let me scroll down just a second. Oh, here we go now. There we go. So the last two that I want to speak about with regards to helpful bacteria is the idea that they help to produce medical compounds, specifically antibiotics as well as other things. And they are also, these antibiotics are used to inhibit the growth of microorganisms. We spend quite a bit of time talking about it in the next lesson when I speak of part 2, but also throughout the course as we move into some of the more medically related fields of biology. We also utilize them with regards to genetic engineering. We spoke a little bit about it in last lesson with viruses, but we use bacteria, specifically a certain type of bacteria, to produce things like insulin and growth hormone that people may not be able to produce as a result of a disease that they may have. So, you know, there's a multifaceted medical field with regards to genetic engineering and prokaryotic organisms, and we will spend quite a bit of time eventually talking about it later on. And then lastly, the commercial uses of prokaryotic organisms. These bacteria help for producing yogurt, cheese, soy sauce, chocolate, yeast for bread making, anything along those lines, fermentation for alcoholic beverages, all of those commercial uses that yeast and bacteria specifically can help produce, they're going to do that. We utilize them for that. All right, questions before I move on to the next part. Questions? Everyone's good. One person uses the check mark. I have a feeling I know who it is. Yeah. Shout out to you two who always use this check mark to let me know that you're good. Oh, we got some more people saying this is okay so far. Excellent. Happy days. Happy days. Okay, good. That's a little bit more of a critical mass. That's good to see. So if there are questions, as always, please just let me know. I have no idea if this makes sense or not. I can't see your faces to tell if it is or not making sense. I'm probably going to put together a survey at some point to ask questions like this. But for the time being live in the moment, it's impossible for me to tell unless you let me know. So please do so if you do have any questions, and I'm more than happy to talk about them. So how many questions are going to be on the quiz? Anywhere from 3 to 5. I haven't decided yet in terms of if I make different variations of the quiz and use different different quizzes within the class. It will be within around 3 to 5 questions though. It's worth about 5 marks total for your thinking. So it's not a big one, but it'll be there. Alrighty, let's move on to the physical characteristics of bacteria, specifically just the general idea of them. And there's a lot more information on here that I'll share as we go. But the big one for the sake of this class, as well as in general, as we move through talking about bacteria, I'm going to spend a lot of time talking about the cell wall today. I'm going to talk about some of the other things as well. But the cell wall is the big one with regards to kind of what's important for this lesson and lessons beyond. Because the cell wall structure made out of capsule, made out of peptidoglycan, which is the big one for later on, and the plasma or cell membrane. This cell wall is responsible for keeping things out, for keeping things inside of the bacteria, and eventually, as we learn about antibiotics, it's going to be the thing that antibiotic targets. So it's important that we understand the general anatomy of and in terms of the physical characteristics of bacteria with regards to the cell wall. So I'd strongly recommend you take some notes with this. I'd strongly recommend you write it down if you're doing that, type it out, whichever one makes you feel good or helps. I would strongly recommend taking that. Alrighty, so the next part, also I just want to make note that this is just the standard general morphology of bacteria. Not all bacteria adhere to this general structure. The thing that is similar with all bacteria though is the cell wall, general cell wall structure, and I'll talk more about that in the next coming slide. or the next page, sorry. All right, so the highlighted in red part here, let me just highlight it again. These are what's called pilus or pili, and they are responsible for some for movement, along with the flagellum, but they're also responsible for absorbing different things, whether it's nutrients, fluids, DNA, etc. And I will talk a little bit more about them as I move on as well. And so again, it's interesting or important to note that when we think about the DNA that's inside of a bacterial cell, it's very important to recognize that there's two components that we need to consider, okay? The first is that this bacteria's DNA does not have a nucleus, okay? So there's no nucleus surrounding that DNA inside of the bacteria. It's just the cytoplasm, ribosomes, a couple of other organelles, and then the DNA or genetic material. There's no nucleus, and the entire bacterial cell is considered to be the nucleoid region where DNA can exist. Because on top of that DNA that belongs to the cell, we have what's called plasmids, or small regular pieces, of that's not what I wanted, of DNA, that small circular pieces of DNA that exist within that bacteria that may or may not originally be part of its genome. And I'll talk more about that when we talk about bacteria's reproducing, okay? All right, take a second if you're writing this down to write it down. I know you have access to this note. It's called Lesson 5. It's the PDF version of Lesson 5 filled in for your own notes, but I would strongly recommend writing some of this stuff down, specifically when it comes to the cell wall and some of the DNA stuff. And then if you have some questions, you're more than welcome to let me know. Is the blue the cell wall? It's a component of the cell wall, yes. That's the capsule. The cell wall, if you can see here, let me get, these are the three, hold on. These are the three layers of the cell wall, right? And this makes up the entire cell wall. All three of those layers make up the cell wall. Maybe I'll do a little bit better. So here's one layer, two layers, three layers. All right. Does that make sense? Excellent. Again, shout out to y'all who give me that check mark. Thank you. I appreciate it. All right. Hopefully you've written down the important stuff with regards to it. And if you have some questions, we can go over it. Because importantly, I want to really, like I said, I want to focus on some of the anatomical features, but I also want to talk a little bit about the behavior and how some of those structures function together and why it's so important that we understand how they function in terms of classifying them, specifically with regards to things, like I said, the cell wall and some of the other things that we'll talk about. So let's talk a little bit about that in terms of the different types of classifications with regards to some of the traits that we've discussed already. So the first way that we will classify bacteria is going to be by their cell wall. And there's three main ways with which we can classify the cell wall structure. And so recognizing that the cell wall is made up out of those three parts, it's important to think about how much of each of those specific components exist for the three different types. Because in the first classification, there is no cell wall, okay? There's no cell wall. And there are very, 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 very few species of bacteria that have no cell wall. And because it has no cell wall, it is less likely to survive the harsh environments that exist for pretty much any living organism, whether it's change in temperature, change in salinity, change in pH. All of those factors can contribute to destroying or killing a bacteria cell that has no cell wall. The one downside, or the one positive, I guess you could call it, of a bacteria that has no cell wall is that it is able to survive any type of treatment. If we were to get a bacterial infection by a bacteria with no cell wall, it would do a very good job at surviving because the medications, specifically antibiotics that we use, target cell walls specifically. So a bacteria with no cell wall would be very good at surviving any type of antibiotic. Alrighty, so the next way that I want to classify a cell wall, specifically bacteria cell wall, is calling it gram-negative, or a thin protein wall. And so here we have this structure, and I will draw attention to the protein, specifically peptidoglycan layer here, because take note, this has a very thin peptidoglycan or protein layer. It's a very thin peptidoglycan layer, and we call this gram-negative. We call this gram-negative. And if we were to do this activity, or if we were to do this lesson in person where we would do a lab, we would stain different bacteria, grow different bacteria, and stain them. And based on the stain color, we would determine if it is a gram-negative or gram-positive. But we can't really do that. So we'll just talk about the theoretical aspects of gram-negative bacteria. And it will turn specifically safranin, when it comes into contact with gram-negative bacteria, it allows for that bacteria to turn pink. So safranin will stain that gram-negative bacteria pink. And it has a lot to do with how thin that peptidoglycan layer is. So this very thin layer of peptidoglycan allows for safranin to turn the bacteria pink. Okay, likewise, with regards to that gram-negative, obviously there should be a one on the other side of the coin, and we call that bacteria gram-positive. So a gram-positive bacteria has a very, very different peptidoglycan layer. And take note, this entire structure here is the peptidoglycan layer. So when you notice the difference between gram-positive and gram-negative, you'll see that it has a very thick protein wall, a very thick protein wall, and we call that gram-positive. And it will turn, as you guessed by my color choice, it will turn crystal violet. The crystal violet will be absorbed by the bacteria, and it turns it purple. So we have gram-negative, which turns pink in the presence of safranin as a result of its thin protein wall or peptidoglycan layer. And then we have gram-positive bacteria, which has a thick peptidoglycan layer, and it turns purple in the presence of crystal violet. Now you might be thinking to yourself, what happens if we use the stain safranin on a gram-positive cell? Nothing happens. And vice versa. If we use crystal violet on a gram-negative cell, likewise, nothing will happen. Okay. So 3 ways to classify cells by their structure of cell wall. And like I said, with regards to some of these concepts, you're definitely going to have to know this, not for your lab quiz, but for your first quiz that we have in a couple of weeks' time. All right, questions? What are sugar chains, and do gram-positives have sugar chains? Yes, but not as, they're not as predominant. They're not as predominant because these sugar chains here, I'll show you the different color. The sugar chains here, they are as a result of having a thin peptidoglycan layer. But we don't really get into too much details with regards to sugar chains in this class. But gram-positive cells, they don't really have them. They have a couple of protein sugar chains, but not as many. And sugar chains are just really for accepting messages from other cells, specifically bacteria that say, hey, get the heck out of my way. Any other questions? Alrighty, judging by the quietness, I'm assuming that everyone is okay for the most part. If you have a question specifically, does safranin have anything to do with saffron? No, but good guess. It does not have anything to do with saffron. It is just, I think it's named after a scientist. I'm not actually sure about what the history of safranin is, though. I just know that it is a pink stain, and it turns gram-negative cells pink. Nah, it's okay. Okay. Oops. So I will move on to the next one if there's no other questions. I'm assuming that there are no other questions. All right. So let's take a look at the next way with which we can classify bacteria, and that is by colony morphology, or how those colonies and cells organize themselves. So when you think about a bacterial culture, right, even though they are that prokaryotic and unicellular, they tend to grow in colonies and hang out in general areas together. And the way with which we can classify it is by understanding our Latin prefixes with regards to shapes and numbers. So the first top part that I've highlighted in purple, when we think about cauci or coccus, we're talking about round or circular. Bacilli or bacillus, rod shaped. And then spirali or spirillum, we're talking about spiral or corkscrew shaped. When I talk about the prefixes, mono, diplo, strepto, and staphylo, they have their own individual associated meanings, whereas mono is singular, diplo is in pairs, strepto is in strings, and staphylo is in clumps or clusters. Oh, question. Do we have to memorize all of this? Yeah, you should definitely know this, yes. You'll definitely need to know this at some point. But I don't want you to memorize every single individual one. I just want you to know the prefixes and suffixes, because if you know the prefixes and suffixes, that's all you need to know, because you can put them together in any way, shape, or form, right? So if you are able to just know what this is, those highlighted in purple, you'll be able to fill in that chart on your own. But I'm going to go over the chart with you now just to talk about it, okay? So monococcus, mono being singular or one, and coccus or cauci being round. You'll note that I have coccus for all of them. It just makes life easier. But biologists tend to just use coccus, despite the fact that it is the singular term. So when we talk about it as coccus, you don't have to worry about cauci for as you increase in numbers. I really just like the idea of focusing on one specific one with regards to the coccus. So when you think of monococcus, mono being singular and coccus being spherical. Diplo meaning two, coccus meaning, again, spherical. Streptococcus, which is the bacteria that causes strep throat, fun fact, it is a string of round bacterial cells, streptococcus, that create this general structure. And then staphylococcus, if you've ever had a staph infection on a cut, that's a bacteria that tends to form clusters or clumps of spherical or round cells. All righty. Questions for that first group? Can we use these characteristics on our dichotomous key? Yes, definitely. 100%. Strong recommend. Alrighty, let's take a look at the bacillus or bacilli, because as you guessed it, the same thing happens here. Mono, diplo, strepto, and staphylobacilli all means the same thing in the context of bacillus being that rod-shaped, and then mono, diplo, strepto, and staphylo being the orientation of those cells. Again, mono singular, diplo in pairs, strepto in strings, and staphylo in clumps. So if I were to say streptobacillus, you know that strepto is chains or strings, and bacillus are rod-shaped cells. So again, the same naming conventions exist for these two groups. The spirillum is a little bit of a weird one because technically it only exists as a monospirillum. There's no diplo or strepto or staphylo versions of it, but we don't even say monospirillum just because there's only one option for it. So these are the three colony morphology classifications that you'll have to know. When would you use plural? Why are we using the plural? Yeah, we're only using the plural or the singular in this case because it is the only, like for spirillum, right? Or are you talking about in general? Yeah, in general, these two, we really only use the singular version of it just because biologists kind of adopted the naming way or the way that they speak about it. It's kind of just like colloquially spoken about it that way because we never really actually see it in the context of it's never only ever going to be a single cell. And so ironically enough, we call it the singular version instead of the plural version. But it's just like one of those naming conventions that just kind of happened to be after a long period of time. We just call it monobacillus and that we just kind of stick with it. So I'm not going to be too chuffed if you know it as bacillus or bacilli. Really and truly, the big thing is that if I were to say a string of rod-shaped cells, if you tell me it's streptobacilli, that's fine too. That's fine too. Alrighty. So are there any questions about this now before I move on to the next part? Judging by the check marks I'm seeing. Thank you again to the same people that use them. Oh, there we go. More check marks. Excellent. My hope is that if people do have a question, they'll type it out. And if you're not sure about what question you could even ask, that's fine too. This is one of those times where this is just like pretty cut and dry information, right? There's nothing too groundbreaking. It's just you thinking about different types of words as is. Alrighty, let's take a look at the classification of respiration method. And I will spend some time explaining the origins of words, specifically aerobes and anaerobes, because again, our Latin, oh, that don't want green, I want blue. Our Latin understanding of prefixes and suffixes and words in general will help us to understand what these words mean. So when I say obligate aerobes, it must have oxygen to survive. So aero is the Latin word for air. And so aerobe or aerobes are things that need air. And in this case, we're talking about oxygen. So aero is the word for air or oxygen. So aerobes need oxygen to survive. And then anaerobes, the prefix an is without. So obligate anaerobes have to have no oxygen to survive. And even so much so that if they are exposed to oxygen, they die as a result of that exposure to oxygen. So an, without, aero, without air, anaerobes It must have 0 oxygen around it. And then lastly, when we look at facultative aerobes, the entire word of facultative aerobes means that it's pretty flexible. It can survive with oxygen or without oxygen. All righty. So take a second to mull this over, think about it, write some down, write some notes down if you're following along and writing down notes in the lesson, and let me know if you have questions. All right, judging by the lack of questions and the check marks that I see, I can move on in. Okay. So again, when we think about classifying bacteria, and someone's already asked the question, but 100% you should be using or thinking about using some of these characteristics in your lab activity that you will, again, have to submit to me on Tuesday, and you will be quizzed on, so to speak. But it's important to recognize that these classifications are also going to be important later on as we talk more about some other organisms that abide by some of these rules. Because if you've already guessed it, humans are facultative aerobes, right? When you think about how our bodies function without oxygen, we got 3 minutes tops before our cells say, okay, that's the end of that chapter. All right. Now, there's also 2 other ways with which we can classify, and that is with regards to the metabolic processes that bacterial cells go through. So when I talk about metabolic processes, I'm talking about how bacteria get nutrients to survive. And so when we think about our Latin origins and Latin words, the idea of auto means that they make their own food. Oops, that's not what I wanted. Autotrophs make their own food, right? When we say autotroph, they make their own food, and it's usually from inorganic chemicals found in the environment, like carbon dioxide and water for plants, nitrogen and minerals for some other bacteria. But ultimately, they're making their own food. Autotrophic, meaning from within, auto, and troph, meaning their food or nutrients. So they make their own food. Whereas, as you guessed it, heterotrophs need to obtain food. They need to obtain food either from an organic source of matter, living or dead, or once living. And that usually means that they're going to turn that food source into glucose with which that they can utilize to make energy. So 2 ways to classify via metabolic processes, which is how they get their nutrients. The first is autotrophic. They make their own food. And heterotrophic, they must obtain it via eating something. Which one are humans? What are humans? Do we make our own food inside of ourselves? Or we got to go search it out, so to speak? Yeah, we're hets. Hets is a weird way of saying it. Heterotrophic, yes, heterotrophs. Exactly. We need to seek out that glucose in the form of plant and animal matter in order to continue making energy, preferably plant material, but. Alrighty, questions about metabolic classifications, because we're moving on to the last few parts of our lesson with regards to reproductive classifications. So it'll be interesting to consider the reproductive systems and reproductive methods of bacteria. And if there are no questions, I'll let you finish writing it down and I'll move on in a second or two. All righty. So let's take a look at the next and second to last classification of bacteria that we will talk about today. And we'll look at how bacteria reproduce. So there's three main ways with which bacteria reproduce. And the first is binary fission or mitosis, which is an asexual reproduction. There is one parent cell that forms two daughter cells that are genetically identical to that parent cell. So there's no mysteries here. It's just like mitosis, with which you learned last year in grade 10. It makes a perfect copy of that cell. And then those two cells go off to make perfect copies of themselves later, so on and so forth. Okay. This one should make quite a bit of sense. My hope is that you remember it from grade 10 science. And if you don't, the big takeaway here is that they are genetically identical daughter cells, and there's only one parent, therefore it is asexual. That's the big takeaway for binary fission. Binary fission. Okay, the next type is conjugation. And this is an interesting one because it is a process that can be seen as sexual reproduction in bacteria. Now you might be thinking to yourself, well, I thought bacteria only reproduced asexually. Yes, but also no, because in this instance where we have conjugation, that plasmid that I talked about earlier in the anatomical components of bacteria, this plasmid gets shared among bacteria, and it makes a bridge and finds a way to transfer that plasmid from one cell to another. And as a result of its transference, and as a result of it being shared with another cell, we consider it sexual reproduction, because technically there are two parents, right? Two parents that are genetically different, and they create an offspring that is also genetically different. And so that offspring that is genetically different, that daughter cell that has a different genetic sequence after the fact, that makes it sexual reproduction. Okay. Oops. So far, any questions for the first two types of reproduction methods for bacteria? No questions. Okay. Alrighty, so the last method that we'll speak of today with regards to reproductive techniques is what I like to lovingly call the, let me write it out here, the bacon strips method. And usually students remember the bacon strips method because bacon strips, and they can be turkey bacon, they can be vegan bacon, whatever floats the boat down the moat. But I call it the turkey bacon. I call it the bacon strips method because these little fellas here, boop, 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 they look like
bacon strips.
Yeah, there we go. Thank you. Bacon or bacon strips. Thank you. Okay, there you go. It took you all the while to turkey. Turkey bacon, if you prefer a healthier turkey bacon alternative. If you're vegetarian or vegan, you can have vegetarian or vegan tofu-based bacon. It doesn't matter. It's bacon strips all the same. So the reason I call it the bacon strips method, outside of it looking like bacon strips, I also call it the transformation method of sexual reproduction, because it is inherently sexual reproduction in the context of a bacterial cell will take in a fragment of another bacterial cell's chromosome, and it will create a genetically different daughter cell. And much like in our last method, right, that we looked at, whenever we have a genetically different daughter cell, we now are talking about a form of sexual reproduction. Because here we have one parent. This is one parent. This is parent number 2. And if you take note at the offspring, or the daughter cell, we call that DC. If you take note of the daughter cell, or DC, it has genetically different genome now as a result of receiving said bacon strip, or that fragment of DNA from that bacterial cell. So now that that new fragment of DNA has inserted itself into that daughter cell, we call this a form of sexual reproduction. This is a form of sexual reproduction. All right. So add that little extra note in. I added it in there for you, but it is a form of sexual reproduction, much like what we talked about above. So if I were to ask a question, for example, on conjugation versus binary fission, or conjugation versus transformation, aka bacon strips method, you would be able to tell me the general idea of whether it's sexual reproduction or asexual reproduction, and the general way with which those two things happen. That should be your main takeaway from all of that. All right, any questions to the reproductive methods, the 3 reproductive methods that we've talked about? You should definitely know them in general terms, and as well as their names, okay? All right, judging by the check marks, thank you once again to those people using those check marks. I appreciate it. It's not hard to do, is it? It's just like click 2 things and just thumbs up or a check mark, yeah? If it's hard to do, you can stop doing it. I mean, it's more just for tofu bacon. Yeah, tofu bacon's delicious. Well, it's not delicious, but it's definitely not bacon. It kicked you. It's because it hates you. What did you miss? What did you last hear? I didn't even notice you're gone. What did you last see? Did you hear the bacon strips method? Oh yeah, that's it. We're still on that part. I'm just about to move on, so you didn't really miss anything. The bacon makes things genetically different. Don't say that in a quiz answer or an assessment test situation. Don't say the bacon makes it a different daughter cell. Be a bit more specific with what that is. All righty. So the last bacon strips method, it's the bacon, the bacon fragment. Yes, there, much better, much more specific now. All righty, let's take a look at the last thing for this lesson. And then if I'm not mistaken, I think you're done. Yeah, we'll have just the last 10 minutes of asynchronous time. But I just want to spend some time talking about what's called endospore formation. And in endospore formation, some bacteria are able to protect their chromosomes with what's called an endospore, which is a very thick peptidoglycan layer or shield that in unfavorable conditions, which I'll talk about later, these unfavorable conditions trigger the bacteria to say, oh, got to go and hibernate. And it wraps its DNA up nice and safe, and it keeps it safe until the environment around it improves. And so what I mean by environment, I mean the idea of, oops, move this out of the way. I mean the idea of things like wrong temperature, amount of oxygen, amount of food, it's too acidic, it's too basic. So in response to these unfavorable environments, the bacteria will shield up, and it will kind of hang out and wait until those environments pass. And so when you find things like endospore bacteria in fossilized creatures or in fossilized stone and fossilized dirt, you start to see that if you give them the correct and favorable environment, those bacteria from hundreds of thousands of millions of years ago are able to actually come back to life. So it's a fantastic way that this bacteria can protect itself pretty much indefinitely until those environmental conditions improve. All right, so that's the last part of the lesson that I wanted to talk with you all about. On Wednesday next week, we will go into part 2 because tomorrow we have a nice little activity planned. Monday's a holiday. Tuesday, your lab is due and we're doing our lab quiz. And then Wednesday, back to the lessons. So you have 10 minutes now to use that asynchronous time to stick around and ask questions. You can work on your lab activity. You can go to the bathroom, do whatever you need to do. I'll still be here for the next 5 so minutes. Yeah, enjoy the rest of your afternoon if you don't have any questions for me, folks.
Bye.
Bye-bye. Is the lab activity getting marked? No. The lab activity is to help you do well on the lab quiz, which is getting marked. And then you still have to submit the lab activity though, right? Because I use it to check against your lab quiz to make sure that no plagiarism occurred. All right, no worries. Have a good one, folks. Bye, everyone. See you later. Bye, Alicia. Bye, Emily. Have a good day.